Hello, everyone, my dear students and fellow learners. This is my introductory video because I'm going to start my YouTube channel um, named Kiran's Biology. In this channel, we are going to discuss different topics from the field of biology, from botany, zoology, cell and molecular biology, genetics, etc. As well as I'll be very happy to take up any topics which you guys will mention in the comment section. So for today, I'm going to start with a very interesting subject, which is cell and molecular biology. And among that, I am going to give you a very brief overview about one of the important processes existing in the cell and molecular biology, just to give an idea and to start my YouTube channel on a good note. So, hi once again. So, the topic which we are going to discuss is related to something to the central dogma. As you can see, central dogma बायोलॉजी के केस में बायोलॉजी में एक बहुत ही इम्पोर्टेंट कॉन्सेप्ट है जिसके अराउंड हमारा पूरा सेल जो है वो काम करता है और पूरी सेल एंड मॉलिक्यूलर बायोलॉजी बेस्ड है so cell and central dogma hota kya hai basically so it suggests that as you can see in this image that the DNA information from DNA it move forwards to RNA through the process of transcription from RNA to proteins through the process of translation. So this is what central dogma is. That is movement of information from DNA to RNA to protein. And among that, we are going to discuss one process which is called as the transcription. Now let's move on and see what transcription is. And we are just going to see an overview how the transcription process is different in both prokaryotes and in eukaryotes, how the process looks different in both of these types of cells. Now, there is a brief introduction or brief definition about the transcription, which is the process of transfer of information from DNA to RNA, which will encode for a specific type of protein. The protein could be a functional protein or a structural protein, depending upon the requirement of the cell. But the important idea here is that DNA encodes RNA, which will have the information to prepare or to cook a protein within the cell that would be utilized in the form of maybe an enzyme or maybe in the form of a cytoskeleton protein or plasma membrane protein or any type of protein. So that is what basically the transcription is. So if you look at these two types of cell, which are basically um, two groups of cells which will um, be present in all kinds of living organisms existing in this world. So, prokaryotic cell, given example, is from bacteria, and the other one is a eukaryotic cell. So, basic differences, I think many of the biology students, right from the class 7th or 8th, we study biology and we start with the one term which is called as cell, and then we see what are the different types of cells. So we know about these types of cell from way back in our learning process. But here I'm just going to, kind of refresh our memories, jog our memories before we dive into details about the transcription in prokaryotes and eukaryotes. So here there are few things which the process of transcription it differs with respect to prokaryotic and eukaryotic cell. One thing is regarding the location. So, where do we see the process of transcription happening in prokaryotes? It's in the DNA, and the DNA is present just inside the cytoplasm, although it will be, kind of condensed towards a certain location called as nucleoid, but as such, there is no outer membrane that encloses the DNA. So basically, it lies into the cytoplasm. However, what happens in case of eukaryotic cell? Here, the DNA which is ultimately going to code for RNA. It's located inside a nuclear membrane, well protected from the cytoplasm and different kinds of enzymes existing in the cytoplasm. And it's bind with the different types of proteins to make a good structure. Um, so the locations are totally different where the transcription is going to happen because of the difference in the DNA and its location and its protection. Then relation with translation is that the if in case of eukaryotic cell where the transcription is going to happen inside the nucleus, so we do have some additional steps that we need to take care of once the RNA is synthesized to make them transported outside. So we have to do some kind of processing, and also with respect to coding and non-coding regions, if you remember from your earlier classes, that because of that also we need to do a lot of RNA processing. We have to do some capping and tailing. But all these complexities with respect to transcription does not happen in case of prokaryotes. They have a very simple type of genetic material which can be transcribed with the help of simple enzyme RNA polymerase, and then a transcript is produced. There is no like there is only coding regions, and mRNA is produced in a very well-defined manner through a step-by-step process. And then, since it is already existing in the cytoplasm, as soon as the mRNA is transcribed, ribosomes start attaching to it, and the formation of protein is also an ongoing process in synchronity with the process of transcription. This is very interesting. Or you and in eukaryotes, what happens, however, that once it is produced, it has to move out. And then once it is out, with the help of a certain structure, maybe the cap, the ribosomes will have to kind of find it. And then the uh, ribosome complex will be formed and it result into the formation of the polypeptide chain. So now, what else is different? So RNA polymerase, so different RNA types of polymerases, they are also different in prokaryotes and eukaryotes based on their complexity, based on the nature where which kind of RNA they are going to produce. So all those minor details with respect to the structure of RNA polymerase and types of it. So this is how the transcription process is going to be different from prokaryotes and eukaryotes. So I'm going to stop here and then we are going to see in the upcoming video lectures regarding the complex process transcription. First, we are going to see in prokaryotes, and then in after that